Genetic variation. Organisms that reproduce sexually differ from one another. Factors causing organisms to develop uniquely include events that occur during meiosis, the cell division process that produces gametes. Because the chromosomes that carry genes occur in pairs, a gene has two forms, or alleles. During meiosis, the chromosomes making up a chromosome pair come together, and their parts may cross over each other. When this happens, DNA strands are broken and rejoined to form new combinations of genes. This process of genetic recombination mixes genes, contributing to genetic variation. After paired chromosomes come together, they separate. Each gamete resulting from meiosis contains only one chromosome from each pair and, therefore, only one allele for each gene. This segregation of alleles helps further ensure that an organism produces gametes containing varied genes. Moreover, genes located on different chromosomes act independently from one another. That is, the distribution of one gene's alleles to one gamete or another does not affect the distribution of another gene's alleles. Because of this independent assortment of alleles, the inheritance of one trait typically is not related to the inheritance of another trait. However, two genes located close to each other on a chromosome may stay together during meiosis and be inherited together. Using logic. Using context clues to determine the meaning of unfamiliar language involves making a logical interpretation based on what you know about a topic. For example, you know that genes produce traits. Mutations. Over time, organisms show traits that were not present in previous generations. These new traits occur because of mutations, or changes that result when errors occur during DNA replication. In DNA replication, the two strands of a DNA molecule unwind from their double helix and then unzip from each other. Each single strand becomes a template for a new strand of DNA. Nucleotides move into place to form a new strand of DNA that is complementary to the template DNA. The positioning of nucleotides depends on their bases. For example, where the template DNA has bases CATG, the new DNA will have complementary bases GTAC because C pairs with G and A pairs with T. Sometimes a mistake happens, creating a mutation. Mutations can occur during mitosis or meiosis. A mutation formed during meiosis creates a new allele for a gene that can be inherited by future offspring. Epigenome. An organism's genome, or complete set of genetic material, can be influenced by its epigenome, or the set of chemical compounds that affect DNA. These chemical compounds do not change DNA, rather, they regulate the activity of genes. That is, epigenetic marks can turn genes on or off to make a cell function in a certain way. For example, an epigenetic mark in a bone cell may turn off a gene that promotes muscle growth. Unlike a genome, an organism's epigenome can change due to influences such as environmental factors. Specifically, what an organism eats or drinks and what pollutants it encounters can change its epigenome. These changes may be harmless or may have consequences and be passed to offspring. Role of genetic recombination in genetic variation. A child does not look exactly like either parent because the child inherits genes from both parents. Humans, like other organisms that reproduce sexually, are genetically varied. One reason for this genetic variation is the occurrence of genetic recombination during meiosis. With meiosis, a parent cell divides, and then its daughter cells divide to form gametes. During prophase I of meiosis, replicated homologous chromosomes come together, and their parts may cross over each other. At this point, the chromosomes may break apart and rejoin to form new combinations of genes, leading to greater genetic variation in the resulting gametes. The diagram depicts the crossing over and genetic recombination that can occur during meiosis. Genetic mutations. Errors in DNA replication can result in changes in the sequence of nucleotide bases in the DNA of a sexually reproducing organism. These mutations can be classified in various ways, some of which are shown in the table below. Sometimes these changes are to genes, sometimes they are not. A change to a gene can alter the gene's ability to give correct instructions for protein synthesis. Whether a genetic mutation has a negative impact depends on where the mutation is and whether it significantly affects production of proteins. For example, in some cases, a mutation may lead to production of an incomplete protein. Such a mutation can have devastating effects. In other cases, a genetic mutation can go completely unnoticed. Linking environmental factors and complex traits. The advancing field of molecular genetics has given us new tools to use when examining traits in twins. Because they are genetically the same but their environments become more unique as they age, identical twins are an excellent model for studying how environment and genes interact. This has become increasingly important when studying complex behaviors and diseases. For example, when only one identical twin in a pair gets a disease, researchers look for elements in the twins' environments that are different. Data is collected and compared for large numbers of affected twins and coupled with DNA and gene product analysis. These types of twin studies can help pinpoint the exact molecular mechanism of a disease and determine the extent of environmental influence. Having this information can lead to the prevention and treatment of complex diseases. To illustrate, for twin pairs where schizophrenia occurs, in 50% of cases both identical twins in a pair develop the disease, while 10 to 15% of cases in fraternal twins show this pattern. This is evidence for a strong genetic component in susceptibility to schizophrenia. However, the fact that both identical twins in a pair don't develop the disease 100% of the time indicates that there are other factors involved. Mutations in gene switches. Mutations in DNA can occur in genes or in other places, such as gene switches. Mutations to gene switches do not affect how proteins are made because instruction for making proteins comes from genes. However, a mutation to a gene switch can cause changes in where, when, and how much of a protein is produced. A mutation can add, break, or modify a gene switch. When a switch is added, it can make a gene active in a new location. When a switch is broken, a gene may not be expressed. When a switch is modified, it can make a gene more or less active than it normally is, resulting in more or less protein being made. As with certain mutations to genes, mutations to gene switches can lead to dramatic results. Mutations in germline cells. Mutations can occur in either somatic cells or germline cells. When a mutation happens in a somatic cell, it affects only the organism in which it occurs. When a mutation occurs in a germline cell, it can be passed to offspring. Development of new alleles. The DNA in a human cell contains about 6 billion nucleotide base pairs. When a cell divides, it must copy this DNA code. If an error occurs, the cell has the ability to correct the error. An error that goes uncorrected causes a mutation. Most mutations have no effect on an individual. A mutation can have an effect only if it relates to the 3% of DNA that makes up genes. The other 97% of the DNA in a human cell is non-coding DNA. Some mutations have an impact beyond an individual. These mutations occur in a germline cell. After fertilization occurs, the mutation can be copied into every new cell of the growing embryo. If the mutation affects a gene, the result is a new allele, or new version of that gene. The new allele can then be passed on to future generations. Environment and gene expression. The role the environment plays in expression of genes is becoming clearer and clearer to researchers. There is an entire field, epigenetics, devoted to studying the relationship between an organism's environment and its genes. The epigenome is comprised of the chemical compounds that are not part of DNA but affect an organism's DNA. Some ways in which the epigenome directly affects organisms are readily seen. For example, Siamese cats have a specific color pattern, as shown in the picture. Siamese cats are colored this way due to a particular gene for pigment. This gene is affected by temperature, which is an environmental factor, not a genetic factor. The gene is expressed to a greater degree at lower temperatures. When you look at the picture, you can see that the cat has darker fur on its ears, nose, paws, and tail. These areas of dark fur are called color points, and this coloration is caused by the temperature-sensitive gene. The ears, nose, paws, and tail of the cat are cooler than the rest of its body and, therefore, are darker in color. Studying the epigenome. Epigenetics, or the study of the epigenome, offers reasons for both concern and optimism. First, epigenetics has shown that unhealthful lifestyle choices, such as overeating or smoking, can alter the epigenetic tags at the top of one's DNA, thereby affecting related genes. For example, these epigenetic tags can cause genes for obesity to be more strongly expressed or genes for longevity to be less strongly expressed. Thus, in addition to immediate health consequences of negative lifestyle choices, such behaviors can subject one's children to greater risk of illness and shorter lifespans. On the other hand, according to a Time.com article, scientists are learning to manipulate epigenetic marks in the lab. They have learned to fight illnesses by creating drugs that activate good genes and suppress bad ones. The Food and Drug Administration approved its first epigenetic drug in 2004. Since then, the FDA has approved a group of epigenetic drugs believed to work in part by activating tumor-blocking genes made inactive by illness. According to Time.com, by turning these epigenetic tags on and off, we could tell genes that play a role in many diseases to lie dormant. Scientists hope that epigenetics one day will help combat several significant diseases. Early clinical trials of epigenetic therapy have shown encouraging results for patients with certain illnesses of the blood. Such developments suggest that our epigenomes and their impacts on our children can indeed be changed for the better.